Hello. Hello. Ladies and gentlemen, a very warm and joyous good afternoon to one and all. It is an absolute honor and delight to welcome our distinguished guest, our chief guest, Dr. Tanjore Balganesh. I request our director, sir, to hand over a bouquet to our chief guest and welcome him to this occasion. We also have among us our former director, Professor Soumitra Das, and respected members of the NIBMG family, dear friends, and everyone joining us online to this momentous occasion. I welcome you all to the 16th Institute Day celebrations of BRIC National Institute of Biomedical Genomics. Today is not just another day, it marks a historic milestone in our journey. It was on this very day in 2009 that we received cabinet approval for the establishment of NIBMG, setting the stage for cutting edge research in biomedical genomics. Since then, we have grown, evolved, and achieved remarkable milestones thanks to the dedication and brilliance of our scientists and students and also our administrative personnel. To begin our celebrations on a serene and auspicious note, I invite our students, Ria and Srijata, to present a soulful invocation. Om, Om, Om. Sarvesham Swastirбhavatu. Sarvesham Shantirбhavatu. Sarvesham Purnam Bhavatu. Sarvesham Mangalam Bhavatu. Om. Sarve Bhavantu Sukhinah. Sarve Santu Niramayah. Sarve Bhadrani Pashyantu. Ma Kaschid Dukha Bhag Bhavet. Sarve Bhavantu Sukhinah. Sarve Santu Niramayah. Sarve Bhadrani Pashyantu. Ma Kaschid Dukha Bhag Bhavet. Om. Shanti, Shanti, Shanti. Thank you, Ria and Srijata. Now, with immense pride and pleasure, I invite our respected and beloved Director, Dr. Sagar Sen Gupta, to deliver the welcome address and provide us with an overview of our research activities over the past years. Welcome, sir.
Good afternoon, everyone. Uh, it It gives me immense pleasure, happiness that today we have Dr. Balganesh with us. It is always I have, from the time I took over, I always wanted him to come to our institute, and it took some time, but thank you for accepting the invitation and coming to us on one of the most important days in the calendar of NIBMG. Thank you. Also, it gives me very much happiness to have Professor Soumitra Das with us. I inherited this institute after him, and it gives me a sense of great happiness that he is with us to see and help us in our journey forward. He has been there for me every time I needed help. I have called him up, and he has never said no in all these years I have been here. So thank you, Soumitra, for coming. Today, whatever I speak is on the behalf of every member of the faculty, the administrative staff, finance, the contractual staff, and I think I am very fortunate to be standing in the podium and speaking on behalf of all of you. This is not the work which I have done. This is not the work which any individual has done in this capacity. It is the work which all of us have done in the last 1 year. And this is the report, in a sort of report which we would like to give, where the institute has done and how we plan to take it forward. So broadly, let us, before we go forward, we also always have to look back a little bit. NIBMG, or more correctly, the BRIC NIBMG, is the premier national institute which is focused on improving the human health to excellence in biomedical research. And the mandate and the mission which was there on this day, which was signed on the day which mentioned, involved conducting and promoting cutting-edge research in biomedical genomics, providing evidence-based biomedical genomics approach for betterment of public health, create research, education, service, translational networks among all the scientists and researchers for public health, and finally, maintain the state-of-the-art infrastructure for genomics and proteomics research with a goal to deliver genomics knowledge and technology. These were actually taken word to word from the mandate of the institute, which was there, which was made available to the founders. So as we look back at NIBMG in the last 1 year, where do we stand? I will go into things which will be specifics as I go, but if I look at the wagon hill, which Alpana actually has made it, every year she does it, and this year is no exception. In the last 1 year, we have got around 10.35 crores of extramural funding. We have generated more than a huge amount of gigabytes of human sequences. We have had 45 publications. We have enrolled PhD and IPhD included 12 students. We have covered PhD 3. The number of awards, which I will go into details. A number of awards are 9. We have had 31 eminent personalities coming over here and giving talks. We have organized workshops, events, and symposium outreach, a very good number, 23 and 16. And we have performed more than 1,600 genetic tests in NIBMG. This institute, at this point, we have just few days reached the level of 300 publications in inception. Currently, there are fellows who are members of the National Science Academy, State Science Academy. We have Ramalinga Swamy Fellows, DBT Inspire Fellows, and also a J.C. Bose Fellow. The number of EMR grants which have been received from inception is 48. We are currently having 46 EMR grants going on. The number of EMR grants sanctioned in the last 1 year is 8, And of course, I told about the funding. So if we now zoom in into particulars about each aspect of this is the broad overview. So how has been our publication output in the last 3 years? We see that the publication output has increased over a period of time, as we see it here. The average impact factor has probably not declined, has remained the same, but the impact factor for faculty is now reaching almost 10. So that is a good sign, that is, tells us we are on the right track, and the total number of citations also are quite remaining steady. In terms of extramural projects, in the last 3 years, the number of ongoing grants have remained same, slight increase, but number of remained same. The total number of new grants have probably come down a little bit. It has been a hard time getting grants. And of course, the money received from different funding agencies have actually come down quite a bit. And this has been a source of worry. And I constantly tell my faculty colleagues and fellows to come up and write more grants, something which we have to do in near future. Awards and honors, we are very happy to say that this year we have had 3 PhDs awarded: Sudipta Chakravarti, Abhishekta Ghosh, and Avarna Sinha. And 2 of them were from Dr. Shamila Sengupta's lab, and 1 of them is from the lab of Dr. Molinath Acharya. There have been many number of students who have gone to different conferences, and what we have seen, we are very proud that our students have made us proud, and they have got awards in those conferences. Deepshika, Devasmita, Tehseen, Anjali, Anurima, Supratim, Shubhasri, and Ankita. Thanks to all of you, Institute is known all over the country. Now, if we go into another area, there are 4 broad areas on which we can think the NIBMG, the whole building rests. One is the cancer biology, one is a host pathogen interaction, one is a complex disorder, and one is the population genetics, genetic epidemiology, and statistical genomics. And as you know, these are not bubbles which do not interact with each other. They interact with each other, the faculties are willing to cross over, and which has been reflected in our publication and in even some of the grants which has come in the last few years. So how has some of the highlights of the 45 papers which were published in the last 1 year? I am not able to do justice to all the 45 papers, obviously you understand, but a few jhalak, a few of them which will come out in this next few slides. So the first paper I would like to highlight is the multivariant genetic architecture, which revealed the testosterone-dependent sexual antagonism in contemporary humans. This is from the lab of Professor Anulava Basu and Dr. Anushya Chakravarti. And their study focuses on the genetic basis of differences in the sexes in 12 traits, which they have actually listed out in the paper. They investigated the multivariant genetic architecture of the sexes in humans using GWAS summary statistics from UK data bank, found sexual antagonism mainly between testosterone and andrometric studies traits, and provided the shared genetic architecture between sexes acts as a constraint to the evolution and sexual dimorphism, only when the selection is antagonistic and not concordant. So this study advanced the understanding of biology of the male and female multivariant genetic architecture in human, and it got published in a very well-known journal. I would also like to highlight another study from Professor Anulava Basu's lab, titled Deep Learning Insights into Distinct Patterns of Polygenic Adaptation Across the Human Population. In this study, Professor Basu actually developed Rising, which is a deep learning software for identifying the key biological processes underlying key biological processes, features. Rising is efficient and outperforms the existing methods. Applying Rising on the whole genome sequencing of individuals, a global map of polygenic adaptation from different populations were also obtained. And they also identified unique genomic regions under the adaptations in Africans and observed an extensive sharing of genomic regions among non. Then to go ahead and show about, highlight about the work from Dr. Samsiddhi Bhattacharya's lab, which is in collaboration with Johns Hopkins University and came out in Nature Communication. Shamsiddhi and his collaborators actually titled the genome-wide multi-trait analysis characterizes the global patterns of pleiotropy and unique trait-specific variants. So what he developed, he along with collaborators developed a fast assay, which is a method to allow scalable detection of variant-level pleiotropic association across many traits. He identified close to 2,000 or 2,200 independent pleiotropic loci across the genome and found the lead variants to be associated with a median of approximately 6 traits. Overlapping functional annotations also were done, and they confirmed the variants regulating expression in multiple genes and tissues tended to be pleiotropic. The follow-up analysis of 21 trait-specific variants indicated their links to the expression of trait-related tissues for a number of potentially core genes involved in relevant biological process. The important thing is to take, which I'm sure Shamsi is working on it. We had discussions on this in recent past. I would like to also highlight, moving on from statistical genomics and also the trade into the another part, like to highlight the work done by Dr. Mahua Maulik. Neurotropic murine beta coronavirus infection causes differential expression of connexin 47 in oligodendrocyte subpopulation associated with demyelination. In this publication, the involvement of glial proteins, which are called connexins, in chronic demyelination followed by beta coronavirus infection. The loss of connexin gap junction expression occurs only in the chronic stage of viral infection and was characterized by severe demyelination. Decoupling of astroglial and oligodendroglial connection expression results in consistent loss of astroglial and dendroglial gap junctions, which is otherwise important for maintaining the normal myelination. And finally, the reduced levels of astroglial oligodendroglial gap junction expression correlates positively with the extent of demyelination, suggesting a plausible role of glial connexins in viral-induced demyelination. So this is an extremely important finding and has potential implications, especially when we are living, just come out of a pandemic, which we have successfully come out. And it may have implications in future during potential future pandemics. I'd like to highlight a recent publication which came up from the lab of Dr. Nidhan Vishwas, the genomic hallmarks of endocrine therapy resistance in ER/PR positive, HER2 negative breast cancers. What Nidhan and his group has found is the whole genome sequencing on endocrine therapy, which are on this type of breast cancer, and the therapy was generated and analyzed. He generated these data sets, and from there he found out 3 important genes. P53, of course, all of you know, PIK3CA and ESR1, which actually resistant mutation signature was found to be over-enriched in therapy-resistant breast cancer tumor gene. This, of course, will have implication in the therapy of this type of cancer. Genomic instability characterized by high copy CNVs, copy number variations, skeletal alterations, and telomere shortening was identified as a major marker for endocrine treatment resistance in Indian breast cancer tumors. So what Nidhan did was to show a way towards our colleagues in clinical biology, clinical hospitals, that they can be segregated, and maybe this type of way of treatment can be looked upon in a very much more positive angle. I would like to highlight the work from collaborative work from Dr. Molinath Acharya's lab, Dr. Mahua's lab, and Dr. Shamsiddhi's lab, who actually found out a function, who actually did published in PLOS genetics and showed functional investigation such as SCAT5 involvement in glucomatous neurodegeneration obtained from a GWAS in a primary angle closure glaucoma. So what Shamshiddi and his colleagues actually did is to find out a 13 SNP genic region in a gene family called catnap5, which is associated with primary angle closure glaucoma in Indian population. And he went ahead, zoomed in on one of the SNPs, which caused to open openness of the chromatin architecture, the potential long-range interaction motifs, and in vitro enhancer luciferase activity. He actually showed that. Catnap5 knocked out zebrafish larvae was showed to have a battery of ocular and retinal degeneration phenotypes. And this study also functional evidence of a putative catnap5 in visual neurodegeneration. And finally, a work from my lab. Of course, you guys know my lab, most of my lab is in NII. And from there, we actually showed that there are small molecules which can disrupt Bloom-Rad54 interaction, which hampers the tumor proliferation in a colon cancer chemo-resistant models. What we show is that there is enhanced chromatin remodeling by this complex, which contributes to the chemo-resistance in colon cancer. Bloom is present in the promoters of multiple efflux pump genes which throw out the small molecules, the chemo-resistant small molecules, which causes the chemo-resistance and thereby enhances the chemo-resistance due to the chromatin remodeling and subsequently their transcription. Screening of a small molecule library identified 3 small molecules which disrupted the Bloom-Rad54 interaction at nanomolar range. And these 3 molecules, when delivered in combination with the standard chemotherapeutic drugs, prevented tumor growth in xenograft and also allogenic model systems. So if I take the research starting from the papers which we have published, if we want to go in more translational biology, more application-based biology, NIBMG has started to go towards that. So we have filed the 2 patents, one on BLM variants and methods, where the fragment of BLM and its use for delayed onsets or prevention of cancer has been filed. And also, Professor Arindam Maitra and his group has filed a patent for a method for detecting single nucleotide polymorphisms in a biological sample and method for identifying susceptibility to preterm delivery. We have also initiated with some molecules which I had developed in collaboration with TMH, which I mentioned in the previous slide, to use small molecule inhibitors to overcome oxaliplatin resistance in advanced colorectal cancers. And the process of transferring know-how of gene panel to detect oral cancer to Premas Biotech is actually in advanced stage of development. So talking about that, we are now going into areas of newer research because this is past. Now we cannot live in past, as you know. We have to move forward. So talking about the newer areas of research. One of the institutional programs which we have started in the last 2 years, I mentioned it last year also when I was giving this talk, was a pan-institutional program which tries to understand the alternative splicing landscape during the human brain development. We, as a number of groups have come out, except Mohua, none of us are really neurobiologists, but we have pulled in our thought processes to conceptualize this program. And we have also collaborators from NIMHANS and ISAR Pune. So how is this program has been developed? As you know, the alternative splicing and alternative splicing is an important facet of gene transcription, gene regulation, and brain development actually happens even starting when from utero to almost 20 years of age, after which the brain development almost stops. Alternative splicing occurs at a very high frequency in normal brain tissues, starting from very early stages of our life, and contributes to every step of development of the nervous system, and of course later on in a system biology approach to almost everything what we do on an everyday basis. The precise timing of splicing switches which underline these mechanisms are not so well known. So, and there are a number of, if the development process is shunted or abnormal, developmental related diseases are known, and some of you must be knowing examples of such individuals which have these diseases. So we thought that we will put in a program trying to understand the alternative splicing, the role of alternative splicing using brain development, and how the process is regulated using the genomics and ultimately the multiple omics approach based techniques. The objectives was how alternative splicing events regulate the gene expression profile during brain development and continue and contribute to its regional and temporal cell type specificity. Also, we wanted to investigate if there is any sex-specific differences in the alternative spliced isoform patterns in brain development, which may contribute to gender biases in several brain disorders. So with this thought process, we set up our primary collaboration with NIMHANS, and we have obtained 42 samples total from male and equally almost divided between the male and females. This is the red dot and green dot tells us from which age we have got the samples. The sample range age from 5 months to almost 75 or 80 years of age. We have got the samples from 3 selected regions of the brain, prefrontal, hippocampus, and cerebellum. For a subset of other of other individuals, we've also got the cerebrospinal fluid. And our aim is to do the whole tissue analysis, transcriptome, whole genome analysis, lipidomics, and proteomics. We also are going to do targeted proteomics. We're also going to do single cell and single nuclei analysis. And also, ultimately, the data we will try to do proteomics data integration. So till now, we have actually finished the samples for whole WGS in Oxford Nanopore. We have done the bulk sequencing, and this is a little bit older data, but I think we have now more than 3 number of single nuclei RNA sequencing. So what are the highlights of our present findings? And this is just a very preliminary data which we have got. And expression quantitation identified the known transcript isoforms and almost 42,000 novel previously uncharacterized isoforms in the prefrontal cortical tissues. We have started working on one of the areas. We cannot work on so much. All the PIs have their own programs. We have started working on one area. So linear modeling identified almost 2,000 transcript isoforms associated with age, with isoforms of genes involved in synaptic plasticity, neurotransmission, which exhibits shifts between toddlers and elders. Very distinct shifts are seen, which happens between the toddlers and elders because of these novel transcripts. And similar isoform switches were identified between male-female gender and between different types of genotypes at proximal SNP loci. So this is something very exciting for us, and we are really looking forward to more of the multi-omics integration later on when all the data comes in. I would like to also focus on another part which is now taking off. It is on the verge of taking off because the money has not yet come, but the sanction order has come. Let's put it in this way. This is a title which is called Advanced Pandemic Preparedness, Innovative Multidisciplinary Strategies for Combating Severe Dengue. And in short, we call it COMBAT. So COMBAT is actually an initiative. It has been there in the pipeline. We have been working on it for the last 1.5 years with our collaborators in Europe. And it has front of dengue research, pandemic preparedness, for pioneering innovative antiviral strategies to alleviate the burden of dengue infection, and at the same time deepening our knowledge to understand its complex pathogenesis. We not only want to develop about the strategies, we want to put it forward as documents, as functional biology to the next step of dengue preparedness. And this is funded by European Union's Horizon Research, Innovation and Department of Biotechnology, Government of India. So this is a 4-year project, and this will be taking off as soon as the funds start coming in. These are the lists of participants in that. As you see, it has a very strong European participation in multiple countries. And the important thing of this project I should also mention, this will bring into our institute a number of techniques which we don't have hands-on, like the Oling technique. We will be giving access to that Oling technique. So this is something which we have worked on, and these are the things, many of the things, the chip-on-chip technique, the brain development, organoid brain development in our institute will be part of this program. In India, we are the lead institute along with Regional Center of Biotechnology, Manipal, and also the others who are clinical cohorts. And we also have a collaborator from US. So the next program I would like to highlight is about the One Day, One Genome program, popularly called ODOG, which the name acronym, which actually showcases the enormous microbial diversity and potential of India in harnessing the microbial diversity. And annotated microbial genomic data is being publicly released every day. Just think about it, last more than 2 months, Shobhik and his group are actually annotating, getting the genomic data, in some cases sequencing them, putting it through a developed pipeline, which is calling Bharat pipeline, learning about what are the genes which are there in this or microorganisms, some of them are pathogenic, and putting it up in the website in collaboration, in coordination with other BRIC institutes. This is the Pan-BRIC initiative which we have undertaken. Until now, we have published 73 genomes. Remember, every working day of the week, we are actually publishing one genome. So we are also contributing in different ways to multi-institutional programs, and which, of course, some of them are already multi-institution, but in a formal way, let me put some more into the pipe, into your front of you. All of you probably know that we are part of the INSACOG. I don't have to tell you. This is over 67 genomic sequencing laboratories connecting over 415 sentinel laboratories throughout the country for managing the disease transmission and tracking the genome evolution of SARS-CoV-2. And this has 3 broad areas. One of them is the sentinel surveillance. Second one is the sewage surveillance and hospital network study. This work was started when Professor Das was director here. And BRIC NIBMG, as of now, has actually deposited, is the highest depositor of SARS-CoV-2 genome sequences, 39,540. The numbers are coming down simply because infections are coming down, not doing work. So it is on 13% of all sequences from across the country. So now we are moving forward. We have not thought that, okay, our work is done. We are moving into phase 3 of INSACOG. And what we are doing is the detection of trying to set, we have, in principle, I think this project is also going to be funded by DBT and by RAC, that we are thinking of detecting emerging and new pathogens and monitoring the known pathogens. For example, influenza virus, we talked about it earlier in the day. Microbiome surveillance for monitoring the bacterial pathogens and their antimicrobial resistance. Setting an early warning system for any disease surge and aid in identifying the variants of concern, and it is being funded by the grand challenges of BIRAC, and the money has to come. So this is why I showed the graph. The graph tells the number of grants which have been yes, are high, almost same, but the money which has come is almost half. So that is the reason to understand I am bringing up this thing. We can start only when the funds come. INTIGS is a project which BG is very much familiar with. He is the chairperson of the committee. He is one of the driver forces which actually guides us in this project. The overall goal is to undertake the data-driven research in irrigate TB. The idea is to perform around 32,000 MTB strains from active TB patients and develop a centralized biorepository. The map the genetic diversity of pulmonary and extrapulmonary TB isolates, identify association of mutation to drug resistance. And this work has already started, and BRIC and IBMG has already sequenced 4,117 MTB genomes. Every sample which we have received till today has been sequenced. Another multi-institutional program is the Garbhini program, which is a multi-omics underpinning of the preterm birth. So what is Garbhini program? Actually, many of you know that preterm birth is one of the major causes which is actually slowing down the progress of in India, in a sense that many of the children with preterm birth have shunted growth and developmental disorders. So it cries out for more attention, and what we have got in collaboration with Tristi and RCB, and of course our clinical collaborators from Gurugram, Safdarjung Hospital, AIMS, and MAMC, are actually some phenomenal understanding of the process. We have shown that the genetic variants which predict the risk of preterm birth has been identified in around 6,200 women. Epigenome-wide characterization has been done, analysis has been done during pregnancy to identify the dynamic risk, and bulk and single transcriptome sequencing of placenta at different gestational stages has also been done, and they are in the process of being analyzed and made public to everyone. Not only this part, but also equally important is also the vaginal and placental microbiome, which is being done. And these signatures for early pregnancy, which are associated with preterm birth, is also analyzed and hopefully will be coming out very soon. I would also like to bring out a nascent program which we are starting off in collaboration with ICER, AIMS Kalyani, and of course with TIFR partners, which is part of the Arunda complex. So Arunda has been conceived as a mission to bring together basic biologists and clinical researchers to provide bench-to-bedside solution to tackle the double burden of malnutrition. As you know, malnutrition is a huge problem which is affecting our country and actually slowing down the progress towards Viksit Bharat. So Arunda is the amalgamation of many institutes across, national institutes across the country, as you can see it here. And in a small part of Arunda has come to us because of the presence of our Kalyani cohort, and with the Kalyani cohort, we would like to do, with the help of AIMS Kalyani, a specific project which actually will try to see the association of body composition and sites in non-glycemic, healthy and lean, normal and overweight individuals. Then I will shift my focus towards the project, which is reaching towards its end, but it has given us valuable insight in the genomic and genetic makeup of our whole country. It is called Genome India. Genome India, we collected around 1,600 samples from West Bengal and Jharkhand and recorded the demographics. We have received samples from others from various populations collected by your collaborators. We have generated global screening array data for more than 2,800 individuals. WGS data was generated for more than 2,400 individuals. We have identified the population-specific variation using anthropometric and hand-led DEXA scan for 1,500, 1,600 individuals. And we have shared our data with all our partners, which is across the country. Between the different centers of the project has been done. Data analysis is in progress, and this year we should have the first flagship publication from this. The country should be proud of the Genome India project. National Supercomputing Mission is another mission which we are proud of. The idea is very simple, as you know very much. This will offer all of you who are students who are working here will understand. We need faster data processing, scalable and efficient processing, and seamless accessibility. In this, our institute has actually achieved quite a bit of success, has optimized the enhanced version of MGS analytical tool, has been 6 modules have been integrated into the HPC framework: alignment, sorting, duplicate removal, base quality calibration, variant calling, and CMV calling. This is no mean achievement. We have achieved significant improvement in time complexity. We have HPC enable BWA MEM alignment is now 1.5 times faster, which is now being implemented. Base quality calibration is 1.6 times speed up. HPC enabled sorting module is 1.7 times speed up. And finally, HPC enabled haplotype caller module for variant calling is 1.9 times speed up. This 1.9 times, don't underestimate it. You are actually doing the things double faster. So this is something which is extremely appreciable. Apart from these, there are certain international collaborations, like using small molecules. My lab is trying to understand the basis of synthetic lethality in colon cancer, and the funding is from CEPIPRA. And in the MOMI project, we are funded by the BMGF. They are trying to understand the genomics analysis of the pregnancy outcomes. So what are the core facilities? Now that I have given you a glimpse about the scientific areas, I will move more later a little bit more into the other aspects of the institute, what institute stands for. NIBMG has a number of core facilities, and I am going to tell you about them. So we have the genomics facility, of course, the jewel in the crown. We also have another jewel in the crown, the data center. We have the proteomics facility, human microbiome facility, zebrafish facility. There is a combined facility of multiple instruments here, the FACS, the Bioplex, and a couple of others more, which tells about D and U, and of course the imaging facility. And this together, in the last, from 2024, has accrued more than 184 lakhs for the country. So apart from the usage by all our facility, our own PIs, we have actually have a number of organizations coming and using them from national laboratories, from different universities, and coming to even private organizations. So then we have a new research facility, which is the BSL-3 facility. Some of you may have seen it on the backstage. I will play a 30-second video.
The BSL-3 facility at BritNI has been established under the Department of Biotechnology initiative. It has one virus lab, a bacteria lab, and an animal BSL-3 room. The BSL-3 facility has been designed to work with pathogenic viruses and bacteria. We have obtained all the required RCGM approvals for our BSL-3 facility. We invite researchers to come and use our facility for their research.
So this is done when we started in January 2024, and we finished in 7 months. And now we have actually, the first SARS-CoV-2 virus is being grown, and the first results, I understand from Anup, has come in.
The BSL-3.
So also we have developed, just opposite to us, we have developed the animal house, the experimental animal house, and this person. So this also took us 7 months to start to finish. We have finished it. HOTO has been done, handed over to us, and now we are waiting for a vet to be appointed so that we can go to the different agencies to get the certifications. I am hopeful by the middle of this year, the certifications will happen. NIBMG has hosted a series of scientific events. There were seminars by eminent speakers. Of course, the highlight of the whole year was Dr. Gokhale, Secretary DBT, coming us and telling us about ambition, innovation, and more. There are also other speakers like Dr. Saurabh Dalal, Dr. Arnab Mukhopadhyay, Dr. Siddesh Kamath, and of course, Dr. Satyajit Rath. Many of you must have interacted with him also. We are not only asking speakers to come, we are also nurturing local and national research ecosystem. There has been symposiums with ICER Kolkata. There have been symposiums with AIMS Kalyani, BioE3 policy on cancer research. And we have had a very good interactive environment all through. And tomorrow we have a symposium on infectious biology. I hope all of you will be here, even if you don't work on infectious biology. So we have had very good outreach activities. Again, one of the major areas is the genetic service unit, which we run from SSKM Hospital in Kolkata. And this year we have provided more than 1,600 high quality free genetic tests. We have from 50 different genetic conditions, more than around 1,600 individuals from 2 countries, 6 Indian states, 22 districts of West Bengal, and our graph is going up continuously, except a dip during the COVID time. And this has been led by Dr. Paramita Bhattacharya and has been guided by Dr. Arindam Mitra. That we held multiple trainings and workshops. For example, we have held the Karyashala, this is the DST Serve Accelerated Vigyan workshop. We have held that. It's called Karyashala, if I'm not mistaken. And then NIBMG's 2-day workshop for faculty members who came from different colleges and spent 2 days with us for hands-on training in the premises. The visit by school and college students was also organized. There were 13 such visits. These visits usually happen on Saturdays, and my faculty members have always raised up their hands and actually helped in these visits. Outreach, yes, we open days, we participate in national exhibitions, we go to IASF, very enthusiastic participation from administration, finance, and of course the students. Students go to IASF every year in Science Day. There is a competition, and whoever makes the best model gets the prize to present in IASF. And that is all, we always do that. And of course, the Global Bio, of course, another symposium, conference where we go. Events, there has been multiple events, International Women's Day, Dr. Bushra Atik came, Independence Day, Tree Plantation Drive, there was Hindi Divas, there was Obed Siddiqui Memorial Lecture, Dr. Shantanu Chaudhary gave the lecture this year, and the Republic Day, of course, we celebrate with usual gusto, and we are extremely happy and honored to be having all these events in our institute. So coming back to infrastructure administrative initiatives, none of these would have been possible if there was not complete synergy between administration, finance, faculty, with me just helping the process along. 3 new laboratories have been set up in the 3rd floor, hoping that new faculty positions will open up. So those faculties who will come, hopefully will have laboratories to start working on. For example, the 3rd floor laboratory, Shomitra and Mohua helped hugely in setting up these laboratories, and thanks to them, to put up this sentence. Painting work in the campus has been completed, as you have seen. It looks nice, I feel so, and it looks pretty decent. So there is a dedicated UPS for 3rd and 4th floor has been set up for uninterrupted power supply for the laboratories. Student hostel has been renovated, and 4th and ground floor has been made ready for new students if we can increase our intake. Construction of the main gate is in progress, and 1 SLT new technician is in progress along with 1 assistant professor, which I forgot to put. There are also financial initiatives. They have been GEM compliant. Huge kudos goes out to the stores and purchase, of course, the finance for helping in this. There have been successful integration with the TSA hybrid system in PFMS, and it is running successfully. We have been able to drop many of the paras, and we have implemented e-payment by 100%. Optimal utilization of core grant has been done in all the three major heads the last year, and completed the statutory audit with clean audit report for the last financial year. And there is also efficient management of intramural support for the faculty members. Whatever is done before they put up their hand, it is always there, and thanks to the finance for helping me out in this. So finally, this is my last slide. I have not yet declared the Best Thesis Award for this year, for the last year. The Best Thesis Award for 2024 goes to thanks to you and for putting me out. And I hope there is no question.
Thank you, sir, for your inspiring words and for highlighting our remarkable achievements over the past year. It is quite a boost up for this year. We are honored to have among us Professor Sunil Kumar Khare, Director ICER Kolkata, as our guest of honor for the evening. I request our Director, sir, to welcome our guest for our Institute Day celebrations. It is indeed our privilege today to welcome Dr. Tanjore Balganesh, President of GangaGen Biotechnology Private Limited, as our chief guest for the evening to deliver the Institute Day lecture. I now once again invite our Director, sir, to introduce our esteemed chief guest.
So I am back. So I'm really happy to introduce Dr. Balganesh. I call him BG. And it is a great honor and privilege, and I use the word very sparse. It is a privilege to have you here, BG, and among us. So, Dr. Balganesh was trained in what we know today as Indian Institute of Chemical Biology. At that time, it was known as Indian Institute of Experimental Medicine. Of course, in Kolkata. He speaks and understands Bengali quite well. So he gained, after his PhD, he gained his postdoctoral experience in Brookhaven National Laboratory in New York and in the Max Planck Institute of Molecular Genetics in Berlin. Then Dr. Balganesh also has been awarded an honorary doctorate from the University of Uppsala in recognition of his work on tuberculosis and malaria subsequently. BG came back to India, I don't know exact years, but he will probably in his talk, he will go down the road, and joined AstraZeneca in Bangalore, where he led to deliver AstraZeneca's first anti-tuberculosis molecule for clinical development. An amazing achievement. After working for AstraZeneca for quite a long period of time, BG came out and joined Government of India with a direct letter from the then Prime Minister, Dr. Manmohan Singh. He was the head of a drug discovery program, which was probably too early for its age. And he was a distinguished scientist in that program. He headed that for 5 years as part of the CSIR. He has since then, around 10 years back, after that stint ended, BG joined his present place as the Emeritus Distinguished Scientist in GangaGen. And GangaGen is a unique experiment which has led to foreign investigators in this year investing around 10 million US dollars for drug development. It's a fantastic achievement for leading a small but dedicated team to take the drug discovery program forward against tuberculosis. BG's research experience, of course, as you understand, is in infectious biology, especially tuberculosis, but he has also led diarrheal disease work on diarrheal diseases, broad spectrum antibiotic programs in AstraZeneca. He is in the review panels of many different panels, part of techs of all the he has been part of techs of all the funding agencies. Presently, as I tell INTEX, he is the chairman of that. He continues to guide research programs in many different national laboratories in India. He's a mentor in the true sense of the term. And today, NIBMG is proud to have you here and hoping to learn from you. Welcome, Vijay.
Thank you, Sagar. Thank you, all of you, who've made me feel so welcome out here. I'm privileged to stand before you and talk to you a little bit about what we guys are doing, trying to do, and where we want to go. Sometimes it's more trials and tribulations. Before lunch, I was talking and this I would like to make. Talking to the students, and one lady asked me the question. I don't want to point her out here, but one of the ladies asked me, what will make you recruit me to an industry, to a biotechnology company? What makes the difference, right? I think it's a fantastic question. Nothing is right or wrong, right? But it's, what's the difference? So I went into this, some of you have heard of Pasteur's quadrant. You have Niels Bohr quadrant. Actually, I've forgotten, one of you helped me. And you have Edison's quadrant, and you have Pasteur's quadrant. Pasteur's quadrant is basic research information, which is then funneled towards a translational goal. As I said, that's the aptitude. If I was sitting in an interview, that's the aptitude I would be looking for. So, Sagar's description of the amount of work, the depth, and the breadth, to me is fascinating. And I must congratulate all of you on that. Every one of that is an achievement. So for me, from the other side, then I'd say, how, Sagar, are you going to convert this to maybe a societal cause, maybe a diagnostic, I did see that, and so on and so forth. That's Pasteur's quadrant, okay? All right, so what am I going to talk about? Sagar, just shout when you want me to stop, because what happens is as you get older, you love to hear your own voice. I'm going to talk about, yes. used to the Mac. That's my problem. Okay, so I'm going to talk. I thought I will present to you a completely different approach that we have used in GangaGen Biotech. And I call this non-traditional approach. And what do I mean by that? So the different parts of the talk that I will be talking about, what are they? Okay now? I want to say my own view of the antibiotic era, because a lot of discussion is we have no new antibiotics, the pharmaceuticals have walked away from it, blah, 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 and all that. I want to give you my own perspective on that. And then I will come to what traditional and not, try and define it for you, and then say how it is different and what we are doing. And then I'll talk about non-traditional antibiotic, which we call P128. It's also called staph tame because the idea is to tame staphylococcus infections and where it went and what happened. Before I go into this, a favorite question of mine when long ago when I used to sit in interviews was, what's the difference between an antibacterial and an antibiotic? It's a classic question. The answer is the antibiotic is something which is biologically produced. The antibacterials are synthetic. Now, many of you from a different generation would probably say they're the same. But, you know, the old guys think about it like that. Most of you know this. Penicillin was discovered. The United States produced 2.3 million doses in the invention of novel. Right? This is also a very familiar slide. Excuse me. Can I get some water? Okay. This is the period where brand new antibiotics is coming. Every few years you had, and then all of a sudden we said 2000, nothing is coming. The oxazolidone came. Then 2003, daptomycin. So what's happening? Is it then saying that we went away, we walked away from finding antibiotics, or we thought we, of course, the Surgeon General of the US said around this said that we have conquered infectious disease, right? But look at it differently. Okay? Look at, sorry about the quality of the slide, but 1960 to 2000, these are all derivatives of quinolones. We had nalidixic acid, norfloxacin, levofloxacin, moxifloxacin in 2000. But we still say no new antibiotics from 1960 to 2000. All right? Give you another example. The top panel is all your different penicillins, 1970 to 2000. You had imipenem, meropenem, which is even used today, and you had ertapenem, which is even used today. Then you, in the macrolides, you had azithromycin, which is basically azithromycin, which we use. So it's wrong to say between 1960 and 2000, we did not have antibiotics. What that period was, it was chemical evolution. We had a molecule and we modified it continuously to overcome resistance, and we had new antibiotics. This is the problem where we landed up. Finding new chemical entities became very, very difficult. This whole chemical evolution disappeared. You are not able to make newer ones to prolong that. There were very few starting points. The second phase, which was interesting that, again, with the students is talking about, infection, you take the medicine for 5 days. That is if you follow the doctor's orders. Most of us stop after 3 days. But anyway, 5 days. Chronic use, diabetics, lifelong. So where is the return of investment? In the chronic use. Nothing in the infection. New antibiotic you discovered today, when is it going to be used? When everything else fails. I won't use it until the old ones don't work, which means I find a new antibiotic, I'm going to use it 10 years later. Half my patent life is gone, commercially it's no longer useful. We all know use and abuse. I can ask you how many of you all took the antibiotic for 5 days when you were prescribed. The other point which is becoming more and more obvious is that we need a narrow spectrum, that too much that a broad spectrum does in terms of destroying the microflora. Pricing is always an issue, and clinical trial. I won't have a chance to go through all this, but this is what has happened and changed the scenario of a dramatic need for new antibiotics. But remember, many of us take antibiotics. If you balance it with, will I become all right without taking an antibiotic or take a placebo, you'll see there's a very fine balance. However, most important need for antibiotics is in the cancer area. There, if you don't have our cancer, we won't be able to treat anyway. So what is the overall picture? We had synthetic as well as natural antibacterials, antibiotics. We had this chemical evolution, then this progression stopped. And I don't want to blame the genome, but what happened was the expectation. Around 2000, genome sequences were coming in, and we said, every few months we'll have a new antibiotic. So we've got thousands of targets, we'll be able to find and so millions was invested in genome, and we just had very poor success. Even today, that's the problem. So now this is where I'm going to ask you, can we look at it differently, right? You now have the genome. You know that just looking at single targets using small molecules is not working. How about we say we use this genome-driven discovery into a completely different non-traditional therapy? What is non-traditional? All right? So let's talk about the definition between traditional and non-traditional. Take the classic example of antibiotic like penicillin, right? It has a spectrum, it is for a defined syndrome, it works rapidly, and it can be used as a single monotherapy. That we can call as a classic Fleming discovery, Fleming antibiotic. What about non-Fleming antibiotics? Are there antibacterials with antibiotics which you can call as non-Fleming? Look at these. What about phage therapy? What about antibodies? What about small molecules which have, I'll come to that, different mode of action? Large molecules, microbiome. We club all these as non-traditional therapies. Right? Now let's look at next step. We can look at non-traditional therapy as 4 treatment architectures. And I'll take you, this is a very nice review written by John Rex, for those of you who want to read it. First is what we call as standalone. Standalone means monotherapy by themselves. Phages can be used, which kill bacteria by themselves. Phage encoded lysins. Now, think about the phage, how it works. It infects a bacteria or sort of binds to a receptor, opens up the cell wall, injects its DNA, multiplies, produces its own phage, and then produces a lysin by which it can break the cell wall and come out. So we can use a lysin. It's a protein. We can use it for therapy. Antisense, there's a lot of talk about it today. You can add RNA here. So these are all standalone, right? Then you have what we call as transform, right? For example, gram-positive bacteria, staphylococcus, or many of these, don't have a cell wall which is very complex. So permeability is not an issue. Gram-negatives, your biggest problem is permeability, right? What if you were to add together a molecule which opens up the cell wall, and then your antibiotic can go in. Classic, which is being actually progressed today, is combinations of colistin. Colistin is nothing but polymyxin, punches holes in the membrane, and then you can use an antibiotic which already works on a gram-positive antibody. We call that transform. We then look at a different class. This is well known. This is not new, where you have a beta-lactamase and a beta-lactamase inhibitor. Both can be small molecules. This is being used. Augmentin is your classical example. The last one is interesting, and which is you have something which prevents or works against a virulence factor, which could be something which prevents capsule formation or prevents toxin production, or in many cases, it can prevent many protease from secretion, because proteases are involved in a lot of pathology and keep microbe alive, along with an antibiotic, right? I'm not going to talk about it, but just think back. The issue here is how you progress these molecules, because this molecule can only be progressed in vivo, can't do anything in vitro. All right, so these are archetypes which I call as, not I call, I'm bringing to your notice as non-traditional antibiotics. Important thing here, these two are large proteins. This could be anything, any virulence factor that you're going against. And these are small molecules. You have small molecules and large molecules in this basket of non-traditional therapy. Okay, so then let's move on and ask the question, what's the sort of principles that you worry about? The four archetypes stand alone in argument, novel and difficult. But look here, this group, they're really, really different. They have an absolute new mode of action. However, they will have a narrow spectrum of activity. Lysins are specific to bacteria, phages are specific to specific species. I won't go into this, but you may not have to show superiority. And for example, a virulent, anti-virulence molecule will have no MIC. You can't measure microbial inhibition in your test tube. It works only in the animal. But anyway, they are different. Look, this side is both like colistin, beta-lactamase inhibitor. These are at least one component. One of them may have an MIC, but again, you will have narrow spectrum problems. But these are issues, nobody is denying that. But you can't pass judgment without actually saying, can we get this into therapy? So for example, if you just take phages, and I talk about lysins that we are developing, the biggest issue is that it's a protein, right? You can get immune reactions. Remember that, right? So we got to deal with that. But does that mean one should not try? All right, let's move on. This is a classic. This is now much higher prediction to 2030. We won't worry about that. What I'm then going to talk to you about is what we have been doing. We're actually taking protein antibacterials to address antimicrobial resistance. And I say GangaGen has been doing, design, discover, develop novel proteins. And obviously, it's a step-by-step progression that we try to go forward. Okay. All right, this is only already. What we did was said, okay, what do we want to develop something against? Because remember, one of the things that from a company perspective, and if I want investors, I have to go and say, what is my product going to look like? Once I say that, they're going to ask me, what's the medical need? What's the business? If you have a comparator today, it's selling so much, if you come with yours, why would I buy yours? What's the business? And the last point is, what's the technical difficulty? So I'm going to take you through all of that because we said we're going to go behind Staph aureus, which is a well-known pathogen. But Staph aureus, as a pathogen, we were looking at positioning it for nasal carriers. And I will come to that in a minute, why nasal carriers. All right, so what is P128, or what we call the product as Staph-TIM? We took an enzyme from a phage called phage K and combined it with another domain, which is the lysostaphin domain of a different phage, to make a chimeric protein called P128. And what I want to show in this figure is this is an interesting protein that if you look at phage K, you do an electron micrograph with gold antibodies, you will see that this molecule is actually outside on the tail. So this is called an ectolysins. So we are differentiating ectolysins from endolysins. What's an ectolysin in a phage? It is already a structural protein. When it binds to the bacteria, it opens up the cell wall so that DNA can go in. What's an endolysin? When the phage is multiplied, it makes a protein which opens up the cell wall so that the phage can come out. So here, why we went for an ectolysin. Why did we go for an ectolysin? Remember, this is out in the environment. This phage carries it. It's in the environment. All of us have been exposed to it. So our thought process was, it will probably be less immunogenic because we've already been exposed to it. All right, so we went to P128, and we found exactly where it cleaves. So this is staphylococcal cell wall. If you look at the two strands of peptidoglycan, it bridges, there is a bridge, which is a pentaglycine bridge, and we clearly showed that the molecule cleaves the pentaglycine bridge. It will also cleave if one of the glycines is replaced by serine. Basically, it did a fusion with GST and the SAK fusion protein, took three different molecules, P128, lysostaph, lys16. Lys16 is nothing but just this portion. And showed that while all three molecules will cleave a pentaglycine, only P128 and lys16 will cleave where it also has a serine. Anyway, just telling you what's the mode of action. All right, so then we obviously did hundreds of strains. If you look at time after addition and the lysis, you will actually see P128 doesn't, if you look at Staph aureus USA 300, which is actually a classical strain, within a couple of minutes, it will completely lyse up. As against, it will not touch Pseudomonas, Klebsiella, E. coli, etc. And basically telling you it's very specific for Staph aureus. And then this was a large panel of nearly 1,000 strains, where we showed that it has got very good activity. Just another point, if you look at how this mechanism of action is different from other antibiotics, just look at over time. You have the green one is daptomycin, which is the standard of care today. Vancomycin is the violet line. And you can see, while these things take about 4 hours for activity, P128 destroys it less than a minute. Now, usually this is a problem if this type of lysis takes place with gram-negatives, because you release endotoxin. In gram-positives, that's not a problem. All right? Then we have done, basically you need to do an MIC 90, 105 strains. MIC is around 4 micrograms. And this is another set where it's between 1.25 and 8. It's a protein, so we need to talk in terms of molar. It's about 30 nanomolar when you talk about 8 micrograms. Vancomycin, same thing. MIC is 1 microgram, 700 nanomolar. So heads to heads, if you compare, it's not, this looks like a huge number, but if you try to look at it, it's not too huge. All right. Then we set up a nasal colonization model. What we did was we took mouse, colonized the nasal area with Staph aureus, methicillin-resistant Staph aureus, and then treated it with P128 or treated it with standard of care, which is mupirocin, and we showed that it works fantastic, no problem. Now is when I wanted to say a little bit of background, and I said positioning. If I go to my investor, the question is how and who's going to use it, right? Methicillin, we are talking, this is the work which was done around 2012, 2014, 16, but the work was started before 2010. The trials were done after 2010. Methicillin-resistant Staph aureus was a death knell for anybody who gets that infection, whether it's systemic infection, wound infection, because once it's methicillin-resistant, we didn't have any other antibiotic. Post-2010, I think, around daptomycin came in, orthozoledrones came in, and the picture changed. So MRSA was a ghost. So we said we'll position it for the nasal treatment for the eradication of Staph aureus from nasal carriers. Why is nasal carrier so important? And I'll show you that in a couple of minutes. So this is all published papers. Overall prevalence of S. aureus, colonization, and infection rates in cardiac, orthopedic, and hemodialysis. Just look at the infection rate. This is all Staph aureus, and the percent of MRSA infection in these. Hemodialysis is 42%. So what does it mean? People who carry Staph aureus in their nasal nares, the source of infection is coming from there. This was very well established, not just in this case. If you then look at hemodialysis especially, overall incidence of MRSA population was 2 to 4%. Number of incidents in dialysis patients was 45%. So in 45% of people who are taking hemodialysis, you had methicillin-resistant nasal carriers, which then created the infection. So clearly that was our positioning, saying, okay, we want to take P128, we want to apply it to the nasal nares so that you eradicate the methicillin-resistant Staph aureus. And we are saying it does it so rapidly that you can then go for your dialysis. That's how we had positioned it, okay? What's the business? Okay, before that, my product profile. So what do we do generally for product profile is that we put together a product profile saying our molecule does A, B, C, D, E, F, G. And then we go to your end users, the key opinion leaders, and ask them, if you had a product like this, would you use it? Because I'm not the end user, somebody else is the end user. So we, based on their feedback, then we position, design the product so that it fits that requirement, right? So this is, we called it product X. If you then look, this is all common. What people wanted, that it should eradicate Staph aureus in 30 minutes prior to procedure. Then I use it. Now that's a huge ask. You're saying, I put it there, 30 minutes, I should not have any more methicillin resistance. Then I use it, right? The bar is there. The other guy said, okay, I'll let you use twice daily for 5 days. If you have, also we will have it. So now you've got two product profiles. You've gone to the key opinion leaders, and they've told you how they're going to use it. Based on that, then you ask the question, what's my business need? Are people going to buy it? What's my return on investment? Now, let's look at it. Anybody who wants to use a new one, I should say, how does it compare to the old one? So the old one is mupirocin. It's available now. Even today, you can go and get a gram of it. It was static. It's not cidal. It needs high concentration to show cidality. It takes 5 to 7 treatment twice daily. There's a lot of resistance around. It is trusted and well documented there for many years. It has a specific formulation. It's about $100 per treatment. And this was the market. This is the US market. So now if you compare that to what we were now aiming for, is that it's going to be sided, so it will work much faster. It should have a rapid mode of action, which I've shown you in vitro. Within a few minutes, it can lyse. It has to work in one day. Whether you give it once or twice, it's work in 1 day. It should have a very low frequency of resistance. Our frequency of resistance was lower than 10 to the minus 9. Not a problem. It should have a simple formulation, right? Look at the business case. I won't go into more details, but what both these graphs are looking at is, over a 10, 15-year period. Remember, I told you 2 types of products. One clears in 1 shot, 1 day. The other one, 5 days. So product X and Y are the sort of projections based on the users. It comes to something like with $300 to $400 million. Then we went further into specifics. You go to ICU, patient population who are MRSA, and then what was happening during this period was the UK government, the US government was making it mandatory for decolonization before surgicals were happening. This was the period where then we said, okay, obviously your market is going to go up dramatically. And if that happens, then you have a market of close to $1 billion. So we have a business case, we've got a product profile, we have a novel product, we have patents going all the way 30 years. So it seems to be well set. Let's do the clinical trials. So before that, so the clinical trial, we were talking to US FDA, and we said logically it's easier to go to Singapore, and FDA Singapore is whatever US FDA says, they will agree to that. So their only question was delivery systems, because originally we were going to put it as a nasal spray, and they didn't want that. They wanted you to do it as a swab. So we had to make a hydrogel. They clearly were interested to monitor resistance as we do the trial. They wanted us to do a safety margin study. So it's a topical, nobody really bothers about how much gets into the system, etc. I said, no, we want you to do a full-fledged safety study. So we did that. This is in mini pigs. I don't know how many of you have seen mini pigs. It's a small pig, but it grows very rapidly. So we did tox studies, and basically we had a margin of more than 20, 22, actually finally 50x. What is 50x? Where so in this case, we didn't see any toxicity, but we were limited to go beyond 50x simply because the molecule was not soluble. So then it's called the maximum feasible dose. So the MFD for us was 50x what we actually need, and this was what was allowed. So we did this trial in Singapore, and I won't go into the details of every one of them here, but basically tell you we had 4 parts where we had part A was nothing but safety, healthy volunteers, but 12 patients, but they were all healthy volunteers. This is a single ascending dose. Then part B was again 3 cohorts, 4 patients. This is multiple ascending dose. So we got all the way to 1,000 micrograms per patient. That means 500 per nair. All right, so which were the patients we were now focusing on? I told you has the maximum number of infusions. So we went to renal dialysis patients. And we did 3 times a day. This was, we had problems in recruiting this. See, the problem in recruiting this is when you go to a patient and you say, I want to test it, most of them are not going to give consent. You know why? 2 reasons. One is I don't have the disease. I don't have a problem. Why should I get treated? That's one. 2, if I get treated for as a part of the trial, Even if I want it later, insurance will not pay for it. Because insurance will pay when you have a problem, not when you not have a problem. So we had lots of problems there in trying to recruit. Anyway, then we went to any patient who's carrying methicillin-resistant Staph aureus in the nasal nerves, and that's the part D, right? Actually, it took this to about 75 patients. Never saw a single adverse effect. So anyway, patients nasally colonized with Staph aureus, randomization was 3:1, and then we gave 3 doses, 100, 300, and 1,000, and we took the risk, saying 1 dose, and we'll see how long we get, how quickly we get eradication, and what does the profile look like. Great expectations. I mean, about 5 years of work, lots of patients, blah, 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 1 slide, right? Look at this. Percent change from baseline of colony-forming units of Staph aureus in nasal samples. In A is 30 minutes, and B is 4 hours. And this is placebo, where you get a very nice high level of numbers. You get a beautiful dose dependence. And if you actually look at this compared to this, is very statistically significant. That at you get fewer number of patients, and by here, 90% of the patients are completely eradicated. So our problem in this trial was in the 60 minute when we went in, 4 hours later when we checked. Our whole trial was derailed because of two points. One, the placebo numbers started to come down. So if your control goes wrong, what are you going to do? We all know what we're going to do, right? However, from a feel-good factor, 300 micrograms clearly was still holding, and 1,000 micrograms was. The bottom line was, the primary endpoint was eradication. We did not reach. Bottom line was, can we hold it for 30 minutes? Yes, we were able to hold it for 30 minutes, right? So that's a story which took us about 8 years. A lot of learning. So we started this work in 2005, 2015, the primary endpoint was not reached. All this was actually mostly done in Bangalore and then trial in Singapore. So last 2 slides. So what now? Staph didn't work. What are you going to do? The business need disappeared. 2 new antibiotics came, oxazolidinone came, and daptomycin came. And the most simplest thing that happened was that nurses were made mandatory to wash their hands, and they were being monitored. That dramatically reduced MRSA infection. Now remember, if our infection rate is 2%, and if it goes down by 40, 30, 40%, my normal values is going to be less than 1%. I can't do a clinical trial with that. Because with 1%, I have to do 1,000 patients to get a statistical number. All right? Today, mupirocin is still your standard of care. Pivotin is also being used. Today, last year, Carbex and a couple of other organizations, I was seeing a massive resurgence in MRSA. What do the stakeholders want? Single dose, 3 months protection. So I won't be able to talk about it, but we went back and we now have protection for at least we do some sort of a healthy volunteer trial. We can clearly see protection lasting for more than 10 days, but not 3 months. We still need to work on it. So what does all this learning finally lead us to? And what has happened to GangaGen in terms of, okay, we did all this, we learned all this, where's GangaGen on this? So this is where I want to take your attention. This is very much in line what you guys do, in line but much lower level than what you guys do. Right? So we said, okay, so what is very clear in the phage genome, you actually have licensed all sorts of varieties. And when we started this, maybe there were about 200 phage genomes, but today you know there are lots and lots of phage genomes. Plus, many of these phages are lysogenic, right? So when you do bacterial genomes, you get this. What we found, these licenses have two clear domains, an enzymatic domain and a cell wall binding domain. So what we've been trying to do is identify novel lysine domains, the binding domain and catalytic domain, and we have actually created a library of these lysines, different binding domains, different lysine domains, and we actually have ectolysins now against a number of bacteria, and which can be processed. And of course, now we've also built a bioinformatic tool, so I don't have to sit, not me, people don't have to sit and look at it by the eye, right? Now, all this under the umbrella of non-traditional antibiotics. All right. My last slide. Will non-traditional antibiotics become therapy? The jury is still out. And hopefully, when I talk tomorrow, I'll talk to you about a completely different a non-traditional antibiotic against Klebsiella pneumonia and where we are with that. So we're trying very hard to open up a completely new channel of finding new antibacterials. But I think organizations, number of organizations in India are very good with the genomics, with expression of proteins, looking at, and maybe there could be a concerted effort to look at, remember proteins. When monoclonals came, you had the same problem. Nobody believed it's going to work. Everybody said people are going to die, blah, blah, blah. We've come a long way. We know how to deal with proteins as therapy. Cost is always an issue, but with a better expression, that problem comes down. So I still think when the jury is out, there should be more and can be done investigations on how you can bring non-traditional therapy or non-traditional molecules as therapy. Thank you very much. I hope it is. Are you going to allow questions?
It's very interesting, actually. We saw a whole perspective how this non-traditional ectomycin was developed by you, especially against MRSA. So the question is there itself. You showed in one of the slides, it's acting specifically against SRES or MRSA, but not against other bacteria. What is that specificity caused by? The other domain, binding domain? Or?
So there is this peptidoglycan recognizing domain, and there's the enzyme domain, catalytic domain. Catalytic domain recognizes the pentaglycine molecule. You have two peptidoglycan strands linked by pentaglycine, so it recognizes that pentaglycine. That's why it's specific for staph. Other bacteria don't have a pentaglycine. Many of them have a DLR or D-lactate DLR. That's why it's specific.
Okay.
Right.
Thank you, sir, for that enlightening and inspiring talk and for expanding our views on drug discovery and pharmaceutical industries. I request our director, sir, to felicitate our chief guest as a token of our gratitude and appreciation. I now cordially invite our guest of honor, Professor Sunil Kumar Kare, to the dais to share his views on this special occasion.
It's okay. Can I speak by this? Very good afternoon. I think it's so. I truly feel honored and privileged to be here. And when so my association with the director, Professor Sagar Sen Gupta, is little longer. We worked across the street in two different institutions. I was from IIT and he was in NII. And a time came when IIT Delhi and NII decided to float the joint projects, and we wrote a joint project, and it was successful. The things were very different. We had a um, uh, nanoflower kind of we had an enzyme which could be shaped into nanoflower. This was transglutaminase. One of my PhD students actually worked on this nanoflower creations and got lots of specific and different kind of activities, unnatural too. Uh, and then we found with our other collaborator that it is working against some of the live cancer cells. And then we requested Dr. Sagar Sen Gupta at that time that can you work with us and test these molecules? Uh, and so this project came through. Um, if I remember correctly, there was a small RNA from the other lab, and we put that small RNA along with the nanoflowers and gave to him, and he successfully shown it against cancer cell line. So this is how it was successful. We then submitted further it to DST, SERB to explore it further, and it worked well. I think it's about to close. So this was the association, and then came the time when I got this offer to join here as a director of ICER Calcutta, and only person I knew in this area was Dr. Sagar Sen Gupta. So I was very happy that at least there is a person, and I called him that I will be there and I need lots of help, and it's a new place for me. So he, as a friend and as a professional collaborator also, as usual, always smiling, always saying yes. He said, yes, come. And we were. And our, I think, collaboration is going further. We have a joint now planning to do the joint projects again. MOU is right there. There were some, I think it was hold up for some reason at wrong place, but I cleared it, and very soon we'll have a joint projects together. And what I saw in his presentation today was, you have a microbiome, I think, sequencing, one day, one genome kind, and to me, it appears that gut microbiome is a very interesting thing, which people are correlating with the disease these days. So maybe some of the, and I work on different kind of extremophilic bacteria. So maybe some of them can be useful. But gut microbiome is also very useful system, something we'll explore. But that's apart, I must say that when he presented about NIB, amazing work, cutting edge work, lots of implication for the country, and you are doing terrific science. I think in this part of country, which is little away from DBT itself, which is in center, but the kind of work which is coming out of this place. Other thing which appealed me was, you see his range of collaboration, all sort of wherever it should be, the institutions, the country, he is there. And not only just collaboration. MOUs normally come up and they die also. But in this case, I saw fruitful production. I saw when I came, sorry I came late, but I saw the patent slide immediately, and first patent was his. So, and then many more. So I congratulate you, all of you, your institution, your director, your scientific colleagues and students. You are doing great, and great for the scientific endeavors and also for the country, progressing science by the country. Genomics is naturally the next era, and you are there. And then icing on cake, and I was very lucky to hear the chief guest, Dr. Balganesh ji. Uh, Really. So, yes, the common perception is that there has not been a kind of any antibiotic after 20s, though some people say try marine actinomycetes, not the actinomycetes, but the rare actinomycetes. But nothing came out. Some molecules, some people say nano will work or nanoparticles along with some molecules works better, whatever. But what is shown here is P128, P128, what he showed was actually working. And MRSA, he did not show you the what are the implications. Just one slide he showed where he said it can affect, these are the numbers in the hospital patient. But one of my students recently published a paper on MRSA itself with the University of Buffalo. Collaboration was there again. And when she shows the slide of introductory slide for what MRSA can do and how dreadful it is, it's really what he has done is remarkable to tackle it. So congratulations, and I was delighted to listen to you. And again, I will say that I'm next door neighbor to you, ISER Calcutta. We are basic science institutions imparting BSMS program integrated and also integrated PhD and PhD program in all basic science disciplines. Facilities are okay, but not as good as you have. And while reading the infrastructure here, I saw many things they asked me are here, and they ask we need it. It's here. They should use it. And so maybe I'm just an arm away. Anytime if we can be of any kind of help, support, and collaboration, I'll be delighted. With these words, I wish you all the best and very happy Foundation Day for NIBMG. Thank you very much.
Thank you, sir, for those inspiring and kind words. I request our director, sir, to felicitate our guest of honor, Professor Sunil Kumar, as a gesture of our sincere appreciation. And now, to formally conclude the academic segment of today's celebrations, I invite our Associate Director, Professor Arvind Moitro, to deliver the vote of thanks.
So this is going to be very short and sweet, hopefully. So every good thing must come to an end, and our Institute Day celebration, at least the academic part of Institute Day celebration, today concludes. But it was a fantastic part of the day that we spent from early morning, and the second half particularly with all these introductions and these talks was really fascinating. So it is now formally time to express our gratitude for whom this has happened, and throughout the year, whatever work was done, and that reflected itself in today, whatever was presented. So first and foremost, I express on behalf of the entire institute and the family members of the institute, I express my deep gratitude to Professor T. Balganesh for coming all the way here, sir, interacting with us in the morning. We are fascinated with our faculty interaction that we had with you. You had something to say to each of us on our work. that will make us come to you more and more. And I'm sure that will be very beneficial for all of us. Your lecture on the Institute Day was really very illuminating. This was completely, at least to me, this was very new. And this non-traditional antibacterials and what you presented, I'm sure many of us, my colleagues here, would be listening and definitely something they will be thinking of as well. And over and above, we participate in the INTGS under your supervision and your guidance, and we will continue to do that and hope that that will be a big success for TB Mukt Bharat. Uh, I also take this occasion to thank Dr. Meenakshi Balganesh, your esteemed better half. She came all the way again, and it was really very interesting to interact with her. I had an interaction after lunch, and it's a different kind of interaction, but it was very nice. I would like now take the occasion to thank our director, Dr. Sagar Sengupta. So not only for his Institute Day presentation, because that is something that he did. We call it the State of the Institute Address. But through it, whatever he showed you today, I mean, what we can say, I am sure all my colleagues would be unanimous in saying this, that throughout the year, he has worked very hard. He has worked very hard with each of us, given us a lot of his time. We know that how much difficult it is to spend time and efforts, but he has done that throughout the year. And what you see today is basically a culmination of all that. We are very, very grateful, and we hope that we'll continue to do more and better under his guidance and advice. We now thank our guest of honor, Dr. Professor Sunil Khare. It was very nice of you, sir, to come over and grace the occasion. We had been interacting with ISER for some time, but now I'm sure that under your presence there, our interactions will become more vibrant scientifically, and we'll have more productive collaborations and interactions going on. Yes, 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 yes. And finally, we had with us Professor Soumitra Das. I mean, there I take a pause, which is a deliberate, because when he came in 2018, he took over the institute and engaged with each of us and added many new dimensions to the institute. And many of us, most of us actually prospered under his guidance, what he did. And I think not only our faculty, the entire staff, the students always keep on remembering him for the engagements he did with us, not only in science or administration, but also in cultural activities, in his enthusiasm for doing everything, something new done, something beautiful. And even today, when we do something like that, we always amongst us discuss, Professor Das should have seen this. And we take a picture and I send it to him by WhatsApp. So thank you, sir, for continuing your engagement with us. You inspire us, and many of us are actually fortunate to continue to collaborate with you also scientifically. Thank you, sir, for coming all the way. I now thank all my colleagues in the faculty who had been all together. We work together as a team. We are not a big number, but whatever we are, we always interact closely, try to help each other out. And today, the Institute Day shows that we had been successfully doing that. And then we have our large number of students which are very vibrant. Today, Dr. Balganesh was commenting that I see that the students are very vibrant here. They're very, very interactive after the interactions. So that was something, a message I wanted to pass to the students. So continue doing that. Engage in science, engage in all kinds of activities, and keep the institute flag flying high. Also, all the members of the institute. So we have a large, we do not have a large body, but we have a small body of administrators, financial teams, so manager administration, manager finance, with their teams, with them, the small group of people. They work extremely hard with us, and they keep on ensuring that the institute works very well. We have a cadre of technical cadre, again a small cadre of staff, technical staff, whether it is system administration, whether it is laboratories, and they all work very hard to keep the institute going. And on behalf of everybody, we thank them in this occasion. Last but not the least, we had a small body of people from various domains, faculty, staff, everybody and students who all took some extra initiatives to make this Institute Day happen, this event happen. And we had preceding this, the sports events happening, and yesterday was the annual sports day. And I thank this small group of people for their extra efforts and time that they spent over the last few days in getting this happen, right from the food menu to the sports events to getting the football. And after this, we have a cultural extravaganza outside. The microphones were being tested, and we had to pause it for this event to happen. Thank you very much for coming, and wish you all a great Institute Day. We look forward to the next events that are going to happen now. Thank you.
Thank you, sir. With this, we officially conclude the academic proceedings of our Institute Day celebration. But as you all are already aware, and like sir has also mentioned, this is just the beginning of a long and exciting evening ahead. We invite you all to stay back and immerse yourself in the cultural extravaganza that follows. An evening filled with music, dance, drama, but in a good way, and many unforgettable experiences. And before that, let's take a break and indulge in some delightful refreshments outside the seminar hall. High tea is now being served. Thanks to each and everyone for gracing this event with your presence. Thank you all.